So our next speaker is Cody Cris, a master's student in biological sciences. His presentation title is Insights into Bacterial Virulence from a Deadly Pathogen. Cody.
I'll try to follow that. Hi, today I'm going to tell you a little bit about the work that our lab does to better characterize how a bacterial pathogen is able to cause disease. You see, well, first I want to acquaint you with some information that you might find shocking. Over the last 2 years, we've all witnessed the havoc that deadly microbes can inflict on our communities. Indeed, the COVID-19 virus has killed over 6 million people worldwide, and in the US, nearly 1 million of those deaths. But what you might be shocked to learn is that one of the leading causes of death is diarrheal diseases, which accounted for more than 1.6 million deaths in 2017 alone. Amongst children, there are about 1.7 billion cases every year, and for children under the age of 5, diarrheal diseases are the second leading cause of death, accounting for 1 in 9 of all childhood deaths. One of the major contributors to this high death toll is a pathogen called Shigella, shown here on the left. Shigella are highly infectious bacteria that are spread through food and contaminated surfaces. The worldwide burden exerted by Shigella is estimated to be about 600,000 deaths and up to 165 million cases every year. In the US, Shigella is attributed with about $93 million in direct medical costs, and due to the rise of antibiotic resistance, Shigella has been designated by the CDC as one of the top 18 pathogens requiring attention right now. Unfortunately, there are no approved and effective vaccines against Shigella, and so we need to identify other ways to treat it, which we can do by understanding how it causes disease. You see, Shigella is able to cause disease due to a set of genes encoded in its DNA, and those disease-causing genes are called virulence genes. As shown on the right, the Shigella bacteria in red, when they are outside the body, these genes are normally kept off. But once inside the body, Shigella's virulence genes get turned on, and Shigella begins to invade and colonize the intestinal lining and destroy it, leading to dysentery. What we're interested in our lab is how these genes get turned on. So what do we know? Well, let's zoom in on the DNA of Shigella. Shown here is a simplified schematic of a Shigella virulence gene. When Shigella is outside of the body, such as on your food, these genes are repressed or inaccessible because a silencer protein comes along and wraps up the DNA to block them. But then once inside your body, Shigella produces an anti-silencer that docks onto the DNA and then somehow is able to counteract that silencer to turn these genes on. What we don't yet understand is how that works. My project aims to figure out how this mechanism works and thereby better understand how Shigella is able to cause disease. Our lab has, through various assays and historical experiments, been able to propose 2 possible mechanisms for this activity. One of them is called spreading, and the other one is called bridging. In the spreading mechanism, that anti-silencer gets recruited to the DNA, it clamps down on it, and then it's interestingly able to slide along the DNA. And this opens up that region again, so that way more of this protein can continue to accumulate on the DNA that eventually disrupts that silencer to turn these genes on. So that's the spreading mechanism. Alternatively, this bridging mechanism, which is another idea we have, is it starts off similarly, that anti-silencer gets onto the DNA, but then instead it cross-links or connects to another copy of itself bound on the DNA. This bridge can lead to remodeling of the DNA structure that disrupts the silencer to turn these genes on. My project is aiming to elucidate which of these mechanisms we can attribute the anti-silencing activity to turn these genes on. And I'm doing this using well-established molecular biology and biochemistry methods. And I'll just add that some preliminary data recently actually suggests that both mechanisms are kind of involved. By characterizing how Shigella is able to regulate these virulence genes, we can better understand how it controls its ability to cause disease. But interestingly, the mechanisms of how Shigella is able to control its virulence genes are predicted to resemble the mechanisms that exist in other bacteria, and hence, the outcomes of my research extend well beyond Shigella and may lead to a better understanding of how other pathogens control their virulence genes as well. Ultimately, this work contributes to our fundamental understanding of topics in biology, but it also provides an opportunity to identify novel drug targets. This may lead to the development of therapeutics and strategies to help treat and fight against Shigella and other bacterial pathogens. Thank you.